Welcome back to my YouTube channel. So today we will study the chapter photosynthesis. Now, before starting the photosynthesis, we first need to know how do plants obtain their nutrition. Okay, so actually plants are autotrophic. We all know that plants are autotrophic. Now, what do we mean by autotrophic nutrition? That means any organism will make their own food, will prepare their own food. They will not depend upon any other organism to obtain the food. Okay, for example, a lion usually eats another animal to obtain its food, to get nutrition, but a plant makes its own food, and the process by which this is prepared, the food is prepared, is called as photosynthesis. Okay, now what do we need for performing photosynthesis? What do plants need? There are some essential things. If they are not present, photosynthesis cannot take place. So first is carbon dioxide, second will be water, and third will be sunlight, right? See, these are the main essential things which a plant will need, or any organism will need, for photosynthesis. Let's start with what is photosynthesis, require, and how were these experiments performed by the scientists to prove that these things are essential, and without these the photosynthesis cannot occur. So firstly, photosynthesis is an endothermic process. Okay, endothermic process means, here what do we need by endothermic process? Means energy is made, energy is produced by photosynthesis process. It is a physiochemical change. It is a endothermic process, and it is a physiochemical change, a physiochemical reaction that takes place in the body of the organism which photosynthesize. Now, we need to obtain some proof, okay, for photosynthesis, proof of photosynthesis. Why? Because we need to prove that some plant or bacteria has done photosynthesis. So in that condition, what will we check to prove that photosynthesis has taken place? First is, if starch is produced, starch has been produced and stored, then that means that photosynthesis has taken place. And second, the oxygen release. Okay, so oxygen will be released, there will be air bubbles of oxygen. So these two are the proof of photosynthesis, which will prove that a plant or a bacteria or some organism which has done photosynthesis, that means photosynthesis has been carried on. Now, first, the experiments which we need to study to prove that these are the essential requirements of a photosynthetic process. Let's go on to the experiments. Now, the first experiment that we have studied in our lower classes was that of the variegated leaf experiment. Variegated leaf experiment. In that, what has been done? That there was a leaf which was covered partly by a black paper. Okay, the leaf has been, one part of the leaf was covered partly by black paper and another part was exposed to the sunlight. After few days, leaf means it was attached to the plant. Okay, it does not mean that it was plucked out of the plant. It was attached to the plant, and the leaf half part was covered with a black paper, which means that no sunlight will reach the green parts of the plant, and other half will have the green parts. Now, what happened was, this part which was covered with black paper could not photosynthesize, so no photosynthesis, and this part will perform photosynthesis. So, what were the proofs of photosynthesis? When we checked for the proof, that means for the starch production, for the starch storage, how was the starch storage checked? The iodine solution test was done. Iodine solution. This part of the leaf was dipped in an iodine solution. So, whichever part will have starch with iodine solution gives a bluish black color. Okay, bluish black color. So what we could infer from the bluish black color, if we see any bluish black color on a part of the leaf which was treated with the starch solution, that it has done photosynthesis and there was starch storage in that part. So if we say that this part has done photosynthesis, so on exposure with the iodine solution it will give a blue black color, right? And this part which was covered with the black paper on exposure with the iodine solution will not give the blue black color, which means there was no starch formation, no starch was stored there. So this was the variegated leaf experiment. Now next experiment was done for the importance of carbon dioxide. What was done? There was a bell jar and a plant was placed, right? A plant which had leaves was placed in the bell jar. For example, this is the plant A. Another similar plant B was placed inside the same setup, but here we had placed some KOH solution in a petri dish. This is the KOH in a petri dish. Now what will this KOH do? It will absorb all the carbon dioxide present in the atmosphere surrounding of this plant. So whatever carbon dioxide will be present inside the bell jar, it will be absorbed by the KOH, so plant will not get carbon dioxide for its photosynthesis. And this bell jar is made up of glass material, so sunlight will be there, of course sunlight will be there, and we have poured water in the plant. Okay, so there will be water available, water is there, sunlight is there, but in plant B there is no carbon dioxide. So again we will test for the starch solution, starch present or not, with the help of iodine solution. So in this case, in the B1, we will see that in this case when we pluck out a leaf and test for the starch solution, bluish black color will not be observed, which means photosynthesis has not been done, starch has not been formed. And in case A, there will be bluish black color because here the photosynthesis has been done, glucose has been formed, which will be stored in the form of starch. So starch will be there and blue black color will be occurring. So these are the importance of carbon dioxide and sunlight. So these two experiments we have studied in our lower classes, so in detail, and now here we will study other importance of other things in photosynthesis. So third experiment we will study about the Priestley's bell jar experiment. Okay, Priestley's experiment. Now what did this scientist do? Priestley, again he used a same bell jar. Bell jar means glass jar made of this shape, because it will be easy to cover the plants, sunlight will be there, water will be present, oxygen or carbon dioxide will be present only inside of the jar. Now he placed a plant in jar A along with a candle and a mouse. Okay, in plant B there was only a candle and a mouse. So what he observed was After few hours, after some time, the candle also the flame extinguished because it used all the oxygen and there was carbon dioxide everywhere, and the mice also used all the oxygen. So mice also was dead because now only carbon dioxide was present here, no oxygen was present for the flame also and for the mice also, because these animals use oxygen, and the candle flame will also utilize the oxygen. They do not use carbon dioxide. So after some time they both will be dead. The flame will also is extinguished and the mice is dead. But when he placed the same when he created a same experimental condition, now a mice was placed, a burning candle was placed, and a plant was placed. Now what happened, this all the candle flame continued to glow and the mice was also alive. How can we explain this? Because whatever oxygen these two utilized was regenerated or was replaced by whatever oxygen the plant gave out, because these two gave the carbon dioxide to plant utilized that carbon dioxide, sunlight and water and gave out oxygen by photosynthesis, and this oxygen was utilized by the candle and the mice. So here the importance of oxygen was proved, and it was also proved that the photosynthesizing plant or the plants which do photosynthesis gives out uses carbon dioxide and gives out oxygen. Okay. And Priestley was also a scientist who was responsible for the discovery of oxygen. So he discovered oxygen. He did not invented oxygen. Okay, there is a difference between invention and discovery. He discovered oxygen. Invention means whatever we do not have, it was created, but oxygen is already present in the environment whether we know or we do not. But he discovered, he came to know about oxygen, and he taught us, he made us know about oxygen is present in our surroundings. So he discovered oxygen. Plus, he also showed the importance of oxygen in photosynthesis, and the green parts of the plant release oxygen when they photosynthesize. Now, next scientist came engine house. Okay, engine house. Now, what he did was he repeated Priestley's experiment. He repeated Priestley's experiment in day condition and in night condition to see whether the absence or the presence of sunlight will affect this process or not. So he came to the conclusion that only in the day the mice in the flame could survive, but not in the night. That means the photosynthesis is carried out only during day. So he repeated the experiment in day and night. Then what he did was he made his own setup. Now he did the same bell jar experiment. He made the same bell jar experiment with, so he used a plant and did the same experiment with a plant known as hydrilla. Hydrilla. He used hydrilla to do similar experiment, and what he observed was that the hydrilla produced some air bubbles. Okay, the hydrilla produced some air bubbles, and we all know that here the air bubbles means that oxygen was produced. He saw some air bubbles, and these air bubbles were coming from the green parts of the plant. So he concluded that when a plant such as hydrilla was exposed to sunlight, was given water, and was given proper carbon dioxide, they performed photosynthesis and produced a gas which was oxygen. Okay, So he also performed this experiment in daylight as well as in the dark or in the night. So he saw that at night there was no air bubble produced, but in the light there was air bubble coming out from the green parts of the plant. So he concluded that oxygen was produced by plants such as hydrilla in the presence of sunlight, carbon dioxide and water, and the process was called as photosynthesis. So they do perform these process in which sunlight was an important factor. Okay. Next scientist was Engelmann. Now, what Engelmann did was he took out a prism. Okay, he focused a light prism on the prism, and the prism, it separated all the colors of light, all the wavelengths of light. Okay, we know that our white light has 7 wavelength Vibgyor. So the prism helped to separate all the wavelengths of light. Now these light were made to fall on a algae cladophora. Okay, algae cladophora. Now what he observed was in the presence of aerobic bacteria, and here aerobic bacteria was present. Now aerobic bacteria means the bacteria which will survive in the presence of oxygen. So when he focused the white light through the prism onto the algae known as cladophora, what he observed was the bacteria were present, the group of bacteria deposited were present on places where oxygen was produced or photosynthesis was done, and here every part will do photosynthesis and produce oxygen, but there will be some parts where the blue light and the red light was falling. Okay, so wherever the blue and the red light was falling, there were more aerobic bacteria. See, Vibgyor. Okay, so wherever there was blue light, there were more aerobic bacteria, and wherever there was red light falling, there was more aerobic bacteria. So he concluded that the area where the blue and the red light was falling was performing more photosynthesis and was producing more oxygen, therefore more amount of bacteria were present. Okay, so through his observation, first action spectrum was formed. Action spectrum means whatever action was done. Now here what action was performed? The action of photosynthesis. Okay, here the action of photosynthesis was being performed. So the action spectrum looked somewhat like this. Wherever there was blue light, there was more photosynthesis, and wherever there was red light region, there was more photosynthesis. So this was of blue light and this was of red light. This was, here we can say that the wavelength of light, and here the action was performed, that means the production of oxygen. So more production of oxygen was found in the area where blue light was falling, and little less was found in the area where red light was falling. So this is called as the action spectrum of photosynthesis. And the NCERT writes that it resembles somewhat the absorption spectra of chlorophyll A and B. Absorption spectra of chlorophyll A and B. So what does it mean? That chlorophyll A and B will also work more effectively, efficiently in the areas or in the wavelength of blue and red light. Okay, it resembles, not exactly, but it resembles roughly. Okay, so the next scientist came, von Zach. What he proved? He told that the photosynthesis, when it took place, the result was glucose formation, and this glucose was later stored as starch in the green parts of the plant. So the green parts of the plant had chloroplast, and these chloroplasts had chlorophyll, which is the green pigment. And due to this, the photosynthesis, due to the chlorophyll, the photosynthesis was done in the presence of sunlight and carbon dioxide and water, and glucose was formed, which ultimately produced starch. So the end result or the storage product was starch. That was told by von Sachs. Now, the final experiment was performed, or the finally we came to know about Von Neil, a scientist named C. Von Neil. Okay, he told the importance of water. He told that whenever we use water, or whenever we say that the photosynthesis is performed and oxygen is released, so this oxygen is not coming from carbon dioxide. Okay, this oxygen is coming from water. He told that water is the reason because of which the oxygen is produced. It is not coming from carbon dioxide. So whenever we use any other substance which is a hydrogen donor apart from water, whatever substance that will be present in the hydrogen donor will be a byproduct. For example, he used purple and green sulfur bacteria. Purple and green sulfur bacteria. So these bacteria also photosynthesize. Now they do not use water for photosynthesis. They used a substance called H2S for photosynthesis. Okay, so what was observed was after photosynthesizing, oxygen was not produced, a sulfur gas was produced in this case. So he came to the conclusion that here the oxygen did not came from carbon dioxide. Because H2S was present, carbon dioxide was also present. Okay, but here if the oxygen would have come from carbon dioxide, here also oxygen would be formed as a byproduct, but here we have seen that sulfur was formed as a byproduct. So he came to the conclusion that oxygen is not reduced due to the presence of water. Oxygen was not produced due to the presence of carbon dioxide. It was the presence of water due to which oxygen as a byproduct was produced. Okay, and this was later proved with the help of radioactivity. What was done was, in radioactivity, what was done that in H2O, when H2O was used, oxygen, usually we know that oxygen 16 is there, but here oxygen 18 was used, and the oxygen which was obtained as a byproduct was also having oxygen 18 instead of oxygen 16. So due to the radioactivity, we also proved that water is the oxygen donor and not carbon dioxide. So this was proved by von Neil. So these were the experiments that proved the importance of the oxygen, carbon dioxide, sunlight, water for photosynthesis, and which proved that the byproduct of photosynthesis is oxygen, and we also obtain glucose, which is stored as a starch after the photosynthesis is done. So thank you very much for listening, and hopefully this concept of all the experiments was cleared here. Please like, subscribe and share this video. Thank you.